I'm Lauren Henke, here with my co-host, Dr. Stephen Chun, with another NRG Oncology podcast episode. Today, we're going to focus on one of the hot topics in cancer research right now, which is precision medicine or precision oncology. To help us navigate precision oncology, we are joined by Dr. Wafik El-Deiry, who is the Associate Dean of Oncologic Science and Director of the Legarreta Cancer Center of the Brown University Alpert Medical School in Rhode Island. Dr. El-Deiry is what many of us would call the true physician scientist triple threat, meaning that he has excelled at cancer related research, teaching, and in clinical care.
On a personal level, I want to give credit to Dr. El-Deiry, who, along with our mutual friend Dr. Nelson Yee, urged me to pursue a career in oncology almost 20 years ago. I will never forget having visited Dr. El-Deiry's lab at the University of Pennsylvania on a cold winter afternoon just because I wanted to see the place where one of the molecular pathways I was looking at in the lab had been discovered. And by chance, Dr. El-Deiry was there after rounding on inpatients, and we spent over an hour talking about the manuscript I had just published in his journal, Cancer Biology and Therapy, and what I wanted to do with my career. In fact, he invited me as a contact on this new platform called LinkedIn, and it was my first connection. I have to say that in retrospect, this was one of the key moments in my life where I really got interested in cancer research because a famous, genuinely nice guy took the time to talk to me.
Arguably, one of Dr. El-Deiry's biggest claims to fame is the discovery that the protein P21 is a transcriptional target of TP53. For our audience members out there who don't know what I'm talking about, one of the fundamental ways that growth of cancer cells can be stopped is when a protein called TP53, named for being 53 kilodaltons, activates the production of another protein called P21. This then activates a cascade of events causing the cell to stop growing. Dr. El-Deiry, did I get this right? And why was this discovery so important?
Thank you, Lauren, and thank you, Stephen, for mentioning with your kind words the positive impact I had on your career. Now that you are very accomplished, it's always touching when someone remembers something like that from 20 years ago. I'll say that P53 is one of these very heavily studied genes and proteins. It goes back to 1979 when it was discovered as a tumor antigen. And for the first 10 years, people actually thought it was an oncogene until the late 80s when work at Johns Hopkins by Bert Vogelstein revealed that the world had been studying a mutated version of P53 for all those years, and that P53 is really a tumor suppressor gene. So this was a time when we were asking, okay, P53 is a tumor suppressor gene. How does it work? How does it suppress cancer? So we became interested in this gene, not just because it looked like a tumor suppressor gene, but it also has proven to be the most commonly mutated gene in human cancer. It's associated with mutations across tumor types. It's associated with poor outcomes in terms of disease behavior and also in terms of response to various therapeutics. To begin unraveling how this gene functions, at the time in the early 90s, it was known that it could bind DNA non-specifically as well as specifically. So I was tasked with determining what the P53 recognized in the human genome and was able to elucidate the sequence that it binds to. So the P21 gene was one of the first genes to be linked to direct control by P53. And it was sort of a bunch of pieces of a big puzzle coming together so that P21 was also discovered as a gene or a protein that binds cyclin-dependent kinases. It was really the first universal cyclin-dependent kinase inhibitor, and that it was a regulatory subunit of such kinases. So, what emerged from all of this and why it's so important is that in the early 90s, it was starting to be recognized that p53 responds to cellular stress, it responds to radiation and chemotherapy, and then causes cells to stop dividing or die. So the discovery of P21 for the first time gave a molecular mechanism of how growth arrest occurs after DNA damage in a general way. Before that time, we understood cell division as just something that goes forward, driven by cyclins and cyclin-dependent kinases. And for the first time, we started to understand how to put the brakes on the cell cycle, how it arrests. A lot of work has been done since then, but I think that was the impact. And it's one of those things that has been reproduced thousands of times at this point.
Having spent some time in a laboratory, I've done Western blots, I've done immunohistochemistry, PCR, and other experiments to understand how drugs and other variables might be affecting this P53 and P21 interaction. I think of how many cancer drugs have been developed because of insight into this signaling pathway. I don't know how the selection process works, but I want to ask, have you ever wondered if you are on the short list for something like the DeBakey Award or even the Nobel Prize?
Well, thank you, Stephen, for bringing that up. I'll start by saying there's a long line and many deserving colleagues for these types of awards. I think this is an important discovery and it could be recognized more. And I'll tell you this, for years after making that discovery, I thought it would be the most important thing that I would ever do as an academic physician scientist. That can be depressing. But thankfully, that turned out not to be the case. We talked about P21 as mediating growth arrest that stops cell division, and that's important in treating cancer. But I'm a medical oncologist and I have a little bit of a one track mind. I want to kill cancer cells, and we knew that p53 could activate programmed cell death. And so, with the knowledge and expertise of the P21 discovery, my lab at Penn pursued the discovery of other targets that mediate cell death. So we discovered trail death receptor 5 as a target of p53 that essentially linked p53 to the innate immune system that suppresses cancer and its metastases as well as viral infections. With that knowledge, eventually, again, because I'm a physician scientist and I'm always looking for ways of translating discoveries, I became interested in looking for small molecules that upregulate this trail death receptor 5 pathway. And my lab discovered TIC10, trail inducing compound number 10. The technology was licensed to a startup I founded called Oncoceutics. The drug went to clinical trials by 2014 as ONC201. And it went into a first in human study at the Cancer Institute of New Jersey, and it went into some brain tumor trials up at Dana-Farber and MGH. In the first glioblastoma trial, there were some exceptional responders with these diffuse gliomas, these middle of the brain cancers that really are only treated with radiation. They don't respond to temozolomide or just about anything else. And there was a big unmet need there. And it turned out the exceptional responders were these diffuse gliomas with H3K27 M mutations. Eventually, there were trials looking at just those patients with H3K27M mutations. And in the end, there was a pretty significant response rate of about 28% and a disease control rate among the majority, around 60 or so percent of the patients. Not everybody responds, but that was remarkable in that there really hasn't been any way to treat, or there certainly were no approved drugs specifically for H3K27M mutated brain tumors. I was very excited, actually thrilled, that ONC201, which also came to be known as dordaviprone or Modeso, and the FDA just gave accelerated approval for what is now known as Modeso, and this approval occurred on August 6, 2025. So I've been asking a lot of questions over the last few years, including mechanistic questions, as well as how can we get this drug to work better across tumor types. And this has taken us far and wide, from mitochondrial metabolism to the integrated stress response to cancer stem cells to epigenetic effects. And it's been exciting, as well as, by the way, immune boosting effects on natural killer cells that are recruited to tumors and mediate some of the anti-tumor effects. So there's a lot of work to do, but that is part of what I'm excited about these days and hope to have impact.
I think it's definitely safe to say that you've had impact. Shifting gears a little bit back to the terminology of precision oncology. This is certainly an area you've been involved in long term. And over the years, we've heard things like targeted biologic or personalized medicine and patient-tailored oncology. We think about therapeutic agents such as Herceptin, for instance, for breast cancer, and Gleevec for chronic myelogenous leukemia. But it also wasn't too long ago that this new thing called 5-fluorouracil came out, and it was felt to be a highly tumor-specific chemotherapy. Now we think of that as being more general. When we hear this term precision oncology, is this just another variation on this new set of buzzwords that we've heard over the decades to describe our efforts to better target our treatments to specific biology of every individual patient's cancer and to reduce side effects?
I would say the examples you gave are some of the best known with Herceptin and Gleevec that are targeting molecular drivers such as HER2 amplification or overexpression in various tumor types, not just breast cancer. And Gleevec, I spent many years in Philadelphia and Pennsylvania, and I always have to mention the Philadelphia chromosome, and it took about 40 years between the Philadelphia chromosome to finding the specific chromosomal translocation to identifying, BCR-ABL to discovery of Gleevec to the clinical trials to drug approval. And that has been incredibly impactful. And there have been a number of other targeted agents where response rates, if you choose the patients appropriately based on molecular biomarkers, you really get fantastic results. I think, the story of EGFR mutations and lung cancer, and those have been very impactful. Historically, in the case of lung cancer, if you took an EGFR inhibitor and treated patients with all comers, the response rate was 10% until it was figured out it was those patients with EGFR mutations. And so in precision oncology, there's always been this way of thinking, you want to give the right drugs to the right patients to have the most impact. And in oncology, nobody wants to treat 100 patients and only have 10 respond. We want to have the majority of our patients benefiting by selecting patients so that they do benefit. I will say a little bit more about precision oncology, but since you brought up 5FU, I will tell you there's another side to targeted therapy and differences between individuals. And so, in the case of 5FU, interestingly, there is variation in the metabolism of 5FU, such that some patients metabolize it more than others. The future there is to be able to dose it better based on pharmacokinetics as well as other predictors of its levels and its expected impact versus toxicities. Precision oncology isn't just about the tumors and what makes them tick and having the right drugs. It's also about the host and giving the right dose to patients of specific drugs. Now, I'll turn and say a few words about precision oncology because you asked me to bring it up to the present. Over the last year and a half, I've been fortunate to chair the WIN consortium for personalized cancer therapy. WIN stands for Worldwide Innovation Network in Precision Oncology. And this is a consortium that was started 15 years ago by John Mendelsohn when he was president at MD Anderson. And this consortium has conducted clinical trials in the area of precision oncology, was really among the very first to use transcriptomics to match drugs to molecular signatures. Part of the challenge in precision oncology has to do with how do we assess benefit. Along these lines, I will mention that a number of national studies such as NCI Match and TAPER, which have illustrated the incredible power of targeted therapy when matched to specific molecular drivers, has focused on single gene abnormalities. First of all, precision oncology is the future of oncology, and it's more than single drivers. And this is being proven in terms of benefit in clinical trials. The WIN consortium was involved with the WINTHER trial that used transcriptomics from tumors as well as normal tissue, and the iPredict study. Both of these studies were published in Nature Medicine, have also explored a parameter known as matching score, which essentially means, if a tumor has multiple drivers, if you target more than one driver at a time, you have a higher matching score. And the patients with a higher matching score actually have better survival outcomes. Currently in oncology, when we see a patient, for example, with advanced cancer, we think about standard of care, we think about clinical trials. And I've been trying to put forth the idea that it's not enough to think about standard of care and clinical trial options, but we should also consider precision oncology options for our patients based on the latest available knowledge. And this sort of process is being facilitated by molecular tumor boards, artificial intelligence, matching scores, and continuing evolution and emergence of supportive data.
As the director of the Legaretta Cancer Center, you've taken on the task of leading and growing a relatively new cancer center established in 2020, as I understand it. And I'm certain that you see the challenges in developing a precision oncology program firsthand. What are the major obstacles that you've been seeing in implementing a precision oncology program? And how can NCI cooperative groups like NRG Oncology, for example, and industry partners better collaborate to implement precision oncology more broadly through clinical trials?
And I would say that precision oncology and our continuing discoveries about cancer among individuals across tumor types has continued over the last couple of decades to just bring out the many, many types of heterogeneity as well as tumor evolution that, in a way, is why we are pursuing precision oncology approaches to be helpful to our patients. But this basically, in some way, means these are more rare tumors. There are clinical trial paradigms. For example, the WIN consortium has some concepts, and WIN is a nonprofit and very interested in collaborating, including with cooperative groups, to bring some of this to reality. I would say, in terms of challenges, some of the challenges are challenges all clinical investigators in the field of oncology or cancer research have, such as drug access, interactions with pharma, and how drugs are approved. If you look at how pharma works and is interested in getting certain drugs approved, in their mind, it's not by N of one precision oncology clinical trials. Certainly, the FDA has not shown consideration for such data in drug approval. If you are a drug company with an approved asset or an early asset under development, the N of one precision oncology clinical trial paradigm is actually incredibly efficient and very powerful in finding signal with combinations to open up new indications for the drugs. And part of the challenge for the field is connecting more with pharma, connecting more with the FDA, connecting more with cooperative groups and with colleagues around the country and around the world to talk about precision oncology and the opportunity here to have some impact. And there is a lot of interest in ways of predicting activities, whether it's AI algorithms or a field that I have become interested in as well, this functional precision medicine, which has, it involves making organoids, for example, and discovering vulnerabilities with specific available drugs, even in the absence of the mutations that they were designed for.
Recently, you wrote an op-ed in the Cancer Letter on how America First means investing in cancer research and specifically investing in the National Cancer Institute. There remains a lot of talk about cutting the NCI budget and support for cooperative groups, including NRG Oncology. Could you give our audience a synopsis of that op-ed piece? And I wonder if you could speak directly to the American public and policymakers on Capitol Hill on why NRG Oncology aligns with an America First agenda.
Over the last 7, 8 months, the Trump administration has hit the ground running with many different policies and activities. And a part of what has been going on has involved changes at Health and Human Services, the NIH, the FDA, and the National Cancer Institute. And we started seeing reorganization of the NIH. We started seeing major budget cuts for the NIH and the National Cancer Institute, along with other, I would say, policy decisions that impact on universities around the country, for example, with indirect costs. And these activities have raised some red flags across the board in terms of destabilizing research across the board, not just at the universities, but also at the NIH and National Cancer Institute with their reach through the field. Now, I am one who has been aware of flat budgets going back to around 2004, 2005. So that's 20 years of NIH budgets and NCI budgets that have not kept up with inflation and have been essentially flat. And for those of us who grew up during this time professionally, we knew about the doubling of the NIH budget, which happened from 98 to 2003. And we were very excited about it. And beyond the doubling, with the flat budgets, some things happened, such as NIH grants became very competitive. There was a larger pool of investigators competing for those dollars and for those budgets. And, the narrative at that time and since then has been, well, we were all very fortunate to have a doubling of the NIH budget, but they just didn't plan ahead for the bigger workforce. And by the way, we are funding more grants. It's just that they're more competitive for your average investigator to get them. So the latest happenings this year were an incredible additional stress on what has been going on for quite some time. And so I was motivated to look into the history of US government support of the NIH and the NCI going back to the 1930s. And when I looked into it, it turned out that the US government has had incredibly strong support of the NIH and the NCI, such that in a couple of decades, in the 40s and 50s, the NIH budget went up 1,000-fold. If you look at subsequent years, including through 1971 and the National Cancer Act that was signed by President Nixon, there was a doubling of the budget roughly every 7 years, up and through 98, and then 98 to 2003, there was a doubling of the NIH budget. And so what I recognize that many people, I think, were unaware of, myself included, is that there has been a history of a doubling of the NIH budget going back 65 years in the last century. And I will mention something that I heard Nobel laureate Bill Kalin say early June of this year at a breakfast with a foundation. He basically said before World War II, Germany was the center of gravity for biomedical research and advances and Nobel Prizes. And after World War II, with these incredible investments by the US government and the attraction to the United States by immigrants, including people like Albert Einstein, led to domination in science and technology and biomedical research, cancer research, lots of Nobel Prizes over the decades. And he ends up with a cautionary note that by reducing the budget and making it more difficult for immigrants to be attracted to the United States, and in fact, we're seeing a reverse brain drain, that we're breaking the chain that has contributed to America being number one. So, with regard to the Cancer Letter article, I came down to it's great that our legislators are trying to avoid a 40% cut. Yeah, that's great. I certainly agree with cutting waste and inefficiencies and addressing some redundancies and so on. But in the end, I feel like we have to have a path to restoring appropriate support for the NIH and the NCI if we're really serious about America first, because it is very competitive. I think you may be aware there are currently more cancer clinical trials in China than the United States. And it goes beyond drugs discovered here being tested there. They're now discovering drugs they've learned and are doing very well. And I think it boils down to what does the future look like? Where will the discoveries be made? Where will the drugs be available and approved? And how do we best serve the people of the United States? And there are many, many benefits to America First beyond sort of the impact on patients. There's impact on the economy and so on from this.
There's been a lot of rumors that you're on the short list for the National Cancer Institute director, and you've even publicly confirmed some of this. Are you able to give us any updates on your candidacy and fill us in on the vetting process? This might be jumping the gun, but if you're made NCI director, we at NRG Oncology will technically be reporting to you. What sort of changes might we expect at NRG Oncology if you become director?
So I will say that I've been a candidate for NCI director since December of 2024. I was vetted early on in December and January through someone who had served as part of Robert F. Kennedy's presidential campaign. And then a few months went by into May and June, and I was contacted by the White House liaison office with HHS and had an interview at that time. I would say that the process for appointment of the next NCI director has not been the most transparent. I think what we see every day is a barrage of many, many things happening well beyond NIH and HHS in general that's keeping the administration extremely busy. I personally think the NCI needs to have an appointed director to focus on cancer and the goals of the NCI to help patients and advance research to have impact on our communities. And of course, an important part of this involves clinical trials. Taking a step back, one of the things I have been focused on when you think about an opportunity like NCI director is how do you allocate your national resources? For me, as an American Cancer Society professor, I'm reminded every day by the number of diagnoses of cancer we will have this year, which is over 2 million, the number of deaths that are predicted in 2025, which is over 600,000, and the growing number of cancer survivors, which is approaching 20 million in the United States. So there is an incredible burden from cancer. Everybody knows somebody or has been touched in some way. But with regard to our national effort, and I think this applies across the board, but certainly in the area of clinical trials, it has to do with impact. How are the things that we are doing ultimately impacting on the burden of cancer? I also think that in thinking about clinical trials, there's obviously a great deal of benefit and necessity from collaborating with big pharma. And I realize there are different opinions about this, but I do think one needs to think about the portfolio in terms of, exactly how innovative are the trials. There are many trials that need to be done, but is everything okay to support through our national resources? And I think that's a good conversation to have. I personally think our national investments should be on the most innovative clinical trials, things that are not going to be done otherwise, things that are not necessarily going to be supported by big pharma. And I think we should definitely pay attention to that in thinking about clinical trials. And I think our cooperative groups need to be preserved and supported to continue their work as they work through some of these issues that I have mentioned.
Dr. El-Deiry, thank you for joining us today on the Energy Oncology podcast. And we want to wish you luck, whether at Brown or the NCI or wherever and whatever the future may hold for you.
Yeah, thank you, Lauren. I had major open heart surgery at the end of July, and right now I am just happy to be alive and back to work. I have a lot that keeps me busy. I'm very excited to be director of the Legaretta Cancer Center at Brown University. A lot of great things are happening here. And with respect to NCI feel like my contribution over the last few months has been to interject into the conversation some things to think about, such as where our investments should be. And whoever gets that role and position, I personally think there should be a path forward to restoring that very strong support for cancer research and clinical trials. It's been said we need a Mary Lasker these days to advocate and to get through. And I think it falls upon all of us to not just be aware of where we are, but just how important advocacy is for our government's support of biomedical research and cancer research. It's not something that any university endowment can replace. When you talk about a $50 billion NIH budget, you literally have to have trillions in an endowment to cover that, and it just doesn't exist. There is no substitute for government support. And so I think there's a lot of work to be done, but I think we do need to pay attention about the investment and where we will end up if we're really serious about America first. And I think we should be. Otherwise, we risk decline and loss of that global leadership. So thanks again to both you, Lauren, and Stephen, for having me on your podcast today.
Today, we heard from Dr. Wafik El-Deiry, the director of the Legaretta Cancer Center of Brown University. He spoke about precision oncology, clinical research, and industry collaboration. If you haven't subscribed to our podcast yet, you can follow us on Spotify, Apple, YouTube, or wherever you get your podcasts. The NRG Oncology Group is supported by grant funding from the National Cancer Institute, and content of this podcast does not necessarily reflect the official views of the National Institutes of Health. Thanks everyone for tuning in, and happy clinical trials.